Hi, my name is Steve Olchin. I'm the Senior Manager for Science and Technology Strategy at Molecular Devices. And today I'll be talking about the many and varied workflows of cell line development. And so what is cell line development? We define cell line development as the process of establishing a clonally derived cell line that has been genetically engineered to produce a foreign material, whether that be a protein or a virus, for example, for a stable period of time. And this process is depicted in the visual below, where at a very high level, which begins with a stable transfection of the protein or gene of interest into a cell host of choice. The next stage is a pool enrichment stage, where cells are often selected for higher copy numbers in order to ensure high productivity in the cell line. Following pool enrichment, we begin single cell isolation, which is a very critical step in establishing clonality of the cell line, which has important consequences in terms of purity and quality of the downstream product. The next stage in the process is to verify single cell isolation. So verify that you've isolated a single cell and not multiple clones, and then monitor that clone for growth. And then move into the next stage, which would then be to screen the clones for titer and potentially other quality attributes, and then scale up your candidate clones, your lead clones. Okay. Now, the applications of cell line development it can be used in a variety of different applications. So ranging from recombinant protein production to natural and non-natural antibody production, and then to maybe a little bit less familiar workflows with regards to cell line engineering for research screening purposes. Now, of course, these are very hot topics and new topics, with the advancements made by CRISPR screening, for example, CRISPR tools. And you can see a variety of different applications that cell line development is applicable for. And cell line development workflow solutions can vary quite significantly, and this is what can make cell line development somewhat challenging to understand. So, for example, if we look at a single stage of this workflow, single cell isolation stage, there are a variety of technologies that are available, ranging from automated liquid handling to fluorescent cell sorters to optofluidics and microfluidics and colony picking. And the challenge here then is in understanding which one of these technologies, which has very different advantages and disadvantages, is most applicable to your particular cell line development workflow. And the additional challenge of choosing a single cell technology beyond just the fact that they're very different technologies, and it's maybe challenging to understand which one and how to compare them to each other, is that the single cell sorting and isolation technology that's chosen will have a very significant impact on upstream and downstream workflows. So what I mean by that is that, for example, each of these images shown here achieves the goal of isolating a single cell, but really accessibility to the cell sample differs tremendously. So, for example, in the case where cells are isolated by the wells of a microplate, you have very easy access to the cells. You can add media reagents at any time, take out media reagents at any time. Whereas cells that are isolated by oil boundary, which is commonly found in droplet microfluidics, makes it very difficult to access the cells once they've been isolated. It's very difficult to get media and reagents in that droplet. In this third scenario, we've got cells isolated by restricting their movement in semi-solid media. Now, there is some accessibility here, but there are challenges with that as well. So in addition to sort of accessibility, there are other advantages and disadvantages of each of these approaches. So one of the disadvantages of using microplates is that it disrupts cell communication, and that disruption can impact cell viability and may alter certain cell phenotypes, particularly those that really depend on cell-to-cell communication, which most cells do. The other limitation of this approach is that you really can only screen a limited number of samples, and it's really limited by the plate format, 96 in a 96 well plate at a maximum. We've already talked about some of the disadvantages of the oil boundary in terms of media addition, but what are some of the advantages of this approach? Well, you can screen a lot more clones, a lot more cell samples in this format in a much shorter time frame, and we're talking, millions of cells as opposed to thousands of cells. It also enables discovery potentially of new cell phenotypes through more sensitive detection. That detection reagents are really confined to a very small region, so you get good signal to noise. Also, disrupts cell communication, that's a problem. And oftentimes this type of solution requires very specialized equipment. And until recently, it was often custom home-built equipment. Right now, there's of course some commercially available products out there on the market, but it's quite expensive and quite specialized. In the third case here with semi-solid media, the primary advantage of this approach is that cell communication is maintained. You don't have any boundaries other than this semi-solid media, which just restricts the movement but allows for small molecules to diffuse. So it really provides a balance of throughput and accessibility to the cells when you compare it to the previous two methods. But there are some disadvantages, of course, as well. Diffusion occurs more slowly, which can impact growth. It still is difficult to change media once these semi-solid media components become more solidified. And again, it also may require some specialized equipment to achieve the throughput that is required. Now, that's just one variable and at one stage of the workflow to consider. There are a lot of additional variables to consider when establishing a cell line development workflow, and we're not, of course, going to have time to go into all of that here. But I just wanted to make that sort of disclaimer that we understand that the workflows that we'll present later on will very much depend on these other parameters that we will not go into. The purpose of today's talk will be to give a very basic overview of common manual techniques that are used in cell line development, and then to discuss the trade-offs to consider when automating and scaling up each stage of these workflows. And then finally, we'll get into a little bit about how these stages connect together into the larger workflow. We're really going to focus on three primary stages of the cell line development workflow because that's where we, molecular devices, have the most relevant products and solutions to meet the needs in cell line development. So we'll focus on these three stages: single cell isolation, monoclonality verification, and clonal outgrowth, and then screening for titer or other quality attributes. So let's begin with single cell isolation. So the most common method that's used today, still today, is limiting dilution. It's a very simple but inefficient technique. What do I mean there? So what is limiting dilution? So limiting dilution is just basically you take a cell culture, dilute down that cell culture to a point which you have very few cells at a given, very few cells, and that concentration, when converted to the number of cells per well, will be somewhere on the order of 0.1 to 1 cell per well. And what this leads to is a situation where the likelihood of a single cell falling into a well follows a Poisson distribution. And for example, at 0.1 cells per well, only 9 out of 96 wells would contain a single cell. So it has 9% efficiency. And this is, by definition, what to expect based on this Poisson distribution. And at that concentration, then you can expect a probability of clonality of about greater than 94%. Now, as you increase the concentration of cells, then you do get an increase in efficiency. So for example, at 0.5 cells per well, efficiencies can be increased to 30%, about 30 or so of the wells having a single cell in them. But that actually results in a decrease in the probability of monoclonality because now you have a lot more wells where there's potentially 2 cells in them. And so you get a probability of clonality that's somewhere around 77%. And so there's always trade-offs that are being made then in terms of balancing throughput needs with the need for monoclonality. Okay, and so this next slide tries to depict this in a nice graphical representation where you can see here on the graph on the left that on the left hand side we have the probability of clonality, and that's shown with the blue line here. On the right graph, we have the max number of clones per plate, and that's the green line here. And then we've got our cells per well. And so what you can see here is at the low end, when we have low concentration of cells, we have a high degree of clonality, high probability of clonality, but about 10 or so clones per plate. On the I think it's about 9 clones per plate. And then as we move up and increase our concentration, of course, we're going to get more clones per plate, and that's going to plateau out, but our probability of clonality is going to go down. Now, this red dotted line here is just to indicate a 90% threshold, which is, commonly would be a minimally acceptable probability of clonality moving forward. Now, on the right-hand side, if we take a look at throughput, what we see here is this is a plot of the number of 96 well plates that would be required to screen 1,000 clones. And so at low concentrations of cells per well, you would need a large number of plates in order to screen 1,000 clones. Almost over 100 plates would be needed. As you increase the concentration, of course, you need less and less plates. But as we recall, there's a trade-off to be made, and we get much lower probabilities at these higher concentrations. Now, the red dotted line here is really to indicate a common threshold for determining what can be handled manually versus what's going to require an automated sort of solution. So anything above 40 plates starts to become unmanageable manually, and really you need to consider automation as part of any solution that's going to involve this many plates. Okay, but herein lies the fundamental problem: is that in order to get an acceptable level of probability of clonality, you really need a low concentration of cells per well at 0.1, 0.2. But to get the throughput, you need to maintain manual processes. You really need higher concentrations, and so there's no overlap between these two, and so you're left making a choice between these two options. Okay, so that simple choice of cell concentration in a limiting dilution workflow has a significant impact on the downstream workflow. Why? Because at a low concentration, you're going to need a highly automated system, right? Screening 111 plates, it's going to require a significant automation. And here's an example of a automated workflow that was pulled from a publication that Bayer had given about their automated cell line development system, right? And you can see all of the different components built out in a limiting dilution automated solution. Now, of course, this costs a lot of money. This is quite expensive, but the advantages here, there are many. One of which being that it allows you then to have a high degree of clonality, and it also gives you a shorter timeline. And I'll talk about that in just a second, but roughly 2 to 3 weeks to get to, to screen your clones. So the second option is to go with a higher concentration of cells, and in this case, I've gone with 1 cell per well, and that's going to require 28 plates in order to screen 1,000 clones. And that can be done manually, and so that's a significant advantage. But the problem here is that you have a low probability of clonality, and that's going to require a second round of cloning to be performed. And now you've instantly doubled your timelines, going from 2 to 3 weeks to 4 to 6 weeks, because you have to repeat this second round of cloning. Okay. So how does that second round of cloning improve the probability of clonality? Well, let's talk about that here. So in the option 1, first round of cloning, we have a probability of 0.94. Okay. Now, let's say to where we did the higher, this is acceptable, this is not high enough. So we had the second round, and the probability of clonality remains the same for any given round. It's 0.77 at 0.5 cells per well. However, in order to calculate the probability of clonality, it's not a simple multiplication of these two probabilities. In fact, that would actually give you a much lower number of probability, and that wouldn't make any sense, right? So here's the equation for calculating the probability of clonality based on limiting dilution. And what you see here is that using this equation, that you get a probability of clonality that's roughly the same as what you got at 0.1 cells per well. Okay, but obviously the disadvantages here are that you again added 2 to 3 weeks to your timelines. Okay, so one way to get around this is to add imaging into the workflow, right? So this is a step that can be added to the workflow that not only improves probability and assurance of clonality, but it also does so without increasing timelines. So from the previous slide here, again, we have 34 plates, we're doing it manually. We've got a probability of clonality of about 0.94. What happens when you add an imager? When you add an imager, the imager's ability to detect clonality typically falls somewhere around about 0.9. And this varies depending on the vendor that you're talking about and your workflow, of course, but most of the literature reports the ability to reliably detect single cells with about a 90% accuracy. So now when you plug these numbers into the equation, you can see here that you get a probability of clonality that's higher than in the previous round, right? So everything remains the same except for you remove the second round of cloning, you can add an imager, and now you get a higher probability at the same level of throughput, right? So that's often, one of the first items to add into any cell line development workflow is to include an imager. Not only does it improve the probability of clonality, but it also gives you that assurance. Seeing is believing. Seeing a single cell image gives you the confidence to know that your clone is indeed monoclonal. But of course, there's always trade-offs. As you scale up and change your workflow, there's always trade-offs. And imaging introduces a bunch of new problems that need to be addressed with regards to throughput and clonality. So for example, of course, you need to be considering the resolution of the cell images that are required and balance that against throughput, because the higher resolution you require, the lower the throughput. The field of view of the imager is also going to have a significant impact because if it's not big enough, it's going to require multiple fields of view to cover the entire well to detect that single cell. And then you can run into problems if those fields of view do not accurately line up with one another. Then you may miscount monoclonal wells. Or wells that are not monoclonal as monoclonal, right? So you really want to make sure that the field of view is taken into consideration alongside the stage accuracy. And so if the stage is not accurate and you require multiple fields of view, you can get situations that look like this. This is from Google Maps, where you get an object that looks somewhat familiar but is not quite lined up properly. If you're considering then fluorescence as part of the imaging solution as well, then additional considerations would be sensitivity of the imaging instrument. In many cases, using a fluorescent reporter may not be the brightest option, and so it's really important to consider the sensitivity of the instrument and then the biological impact of fluorescent reporter or any sort of fluorescent dye that you might add into your cells of interest. And so as an example of the trade-offs that must be made, we have performed a case study where we looked at automating single cell detection on one of our imaging instruments at Molecular Devices. And we wanted to automate that single cell detection using a fluorescent dye. Okay, and in this case, we use calcein-AM. And what you can see is we tested 3 different concentrations of calcein-AM. Hopefully you can see the nice bright cells in image A. In image B, they're getting much dimmer. and in C, it's really hard to see anything. Hopefully you can see in the transmitted light images, though, that there's a number of cells present. Okay. So at the 3 different concentrations of calcein-AM, the imager was not able to detect the 0.5 concentration. So that's not really going to work very well. Now, if we look at the other figures here, if we look at the, I think this is probably easiest. If we look at the number of wells in which a colony survived post-cloning, you can see that in label-free, of course, we don't expect any sort of cell death, and so about 75% of all wells grew into colonies. And then with our 1 micromolar concentration of calcein, similar sorts of viability, but then 5 micromolar, we see toxicity. And so that concentration doesn't work either. So in this case study, and in this particular use case, looking at an adherent cell line, actually, we could see that 0.5 micromolar concentration of calcein-AM was not detectable, so we couldn't use it, and 5 micromolar was toxic. So we found a balance, and 1 micromolar was a sufficient concentration for this particular application. Okay. Now again, this data was collected on the CloneSelect imager. Just a little bit about this product from Molecular Devices. It's a rapid imaging instrument. It can acquire an entire 96 well plate, whole well images in 90 seconds in transmitted light. And it's designed to do label-free imaging to monitor proliferation of single cells into colonies. It gives you that monoclonality verification using high resolution imaging of single cells, and then can be customized for both fluorescence and for to be automation friendly, so be able to integrate into a more automated workflow. And you can see an image of that here on the left-hand side. Okay. Now, let's get back into some additional trade-offs that we need to consider now in terms of single cell sorting, throughput, and imaging speed, because imaging can take quite a bit of time. And you saw the CloneSelect imager takes about 90 seconds per plate. That's industry leading. Other systems that are out there on the market can take much, much longer. And so why is that important? Well, let's take a look at the graph on the left here. That on the x-axis is the time required to image a plate. So 2 minutes per plate would be roughly where the CloneSelect imager is at, right? All the way up to 5 minutes to 15 minutes per plate. On the y-axis, it's the hours needed to screen 1,000 clones. And this is based on a concentration of 0.1 cells per well, which will give us the number of plates that would be required then to get to 1,000 clones. And what you can see in this scenario is that, at 15 minutes per plate, it's going to take over 24 hours to image all of the plates to screen 1,000 clones. And so, in fact, this is a process that requires imaging every single day, at least at the early time points. So you wouldn't be able to screen 1,000 clones if the imager takes 15 minutes per plate, because you would need to image 24 hours later, and that's not possible at this time scale. So that's why this is indicated marked as red. Now, orange and yellow here indicate that it's going to take more than 8 hours, a typical workday, in order to acquire all of the plates necessary to screen 1,000 clones. So none of these imagers at this imaging speed are really amenable to a manual process. You really need automation at this scale, right? But you do get a high probability of clonality at this scale, over 0.99, right? And so if you don't need 1,000 clones, maybe it would be acceptable to have an imager that was a bit slower, but otherwise, these are really not going to be acceptable speeds. However, if we take a look at the hours needed to image 1,000 clones per day at a concentration of 1 cell per well, you can see that it's very manageable for any sort of imager to get through all of the plates necessary in that time frame, right? And so it's really a trade-off that you're making, again, which we've talked about before, that you can get do the manual process, less throughput needed, but the probability of clonality will be lower versus high probability of clonality, but much more throughput needed. So oftentimes you'll see that a concentration of about 0.3 cells per well is pretty commonly used because it balances both the need for throughput and clonality. And so at this concentration, it would take about 47 plates in order to screen 1,000 clones, okay? And you'd get a probability of about 0.98 if you include an imager in this workflow, okay? And only at the upper ends with an imager that takes 10 to 15 minutes would you really start to run into an issue in terms of manually being able to handle this. So this might require a little automation, but generally most of the imagers in this space really can deliver on these imaging speeds. Okay. But that's not the only trade-off that is made between throughput and this workflow. And so another consideration is post-transfection recovery. And so what this graph shows is a transfection of a given cell line at day 0. And then on the x-axis is the number of days that those cells remain in culture post-transfection and their viability rate at that time point. And so following any transfection event, you're going to get quite a bit of cell death at the early stages. And then as those cells die off and cells recover and start dividing again, you'll get a recovery phase here. And what these black arrows indicate then is the day post-transfection at which 50% of the cell culture was viable, okay? And I'm using just a rough estimate here based on these blue-red lines. To get to 75% viability, you'd be at day 15 post-transfection. And then I'm just sort of continuing out this plot here, even though there's no data, to say that roughly by day 24, maybe we would expect 100% viability, which is, you know, generally never get 100%, but let's just say, for the sake of argument, that by day 24 you get to 100% viability. Well, what impact does this have on the workflow? Well, if you're only 50% of your clones grow out, you've immediately lost half of your clones, and that's going to double, you know, essentially the time that's needed to screen 1,000 clones, right? And so that's what's being shown here in the graph on the right-hand side is the 3 different viability percentages, 50, 75, and 100% viability at 0.3 cells per well, how long it's going to take to image 1,000 clones. Okay, so let's say, for example, let's look at 2 minutes per plate. Then the low bar here is 100% viability. This is 75%, and this would be 50%. So once you get to the higher time imaging speeds here, then you can see that at 7.5 minutes per plate, you get to this scenario where at 100% and at 50% and 75% viability, you're not going to be able to handle that with a manual process, right? But at 100% viability, you would. And so you can choose the what viability is necessary in order to be able to screen the number of clones that you require, given your sort of manual and automated setup. And the benefit here then would be that if you can screen clones at 50% viability, that's going to, versus 100% viability, that's going to shave 13 days off of your timeline. So you generally prefer to screen as early as possible, if other things, don't go wrong, right? But if you can screen earlier, that's better. It's going to shave a lot more time off. One of the disadvantages of using FACS to do sorting is that it leads to the situation where you are batch sorting cells, which is going to lower throughput. So let me explain what that means. So batch sorting means that we basically seed all of our single cells into all of our 96 well plates. So let's say we're screening 40 plates. So we do all of our single cell sorting at one time. That can take a long time, up to 4 hours, depending on how much cleaning and sterilization needs to be done, and how much sample prep needs to be done for that particular flow system. But it can take quite a while. And then after that 4 hours, each one of those plates is imaged. And in this scenario, I said that it took about 4 minutes to image a given plate. And in order to get through, oh, sorry, I already had some numbers here. So instead of 40 plates, imagine it's 63 plates. But it takes about 4 hours then to get through all of these plates. Well, in that 4-hour time frame, by the time you get to plate number 63, many of the cells may have already divided by the time you get to plate 63. And this is a problem because you want to capture an image of a single cell. If that single cell is divided, now you have 2 cells before you capture an image, you won't know whether or not that well started monoclonal and then just and divided before you captured an image, right? And so that 4-hour delay results in a loss out of 1 out of every 6 clones in these plates, right? And it gets much more significant if the imaging speeds are slower than the slow sorting speed. So, for example, if it takes 15 minutes per plate to image a 96 well plate, then you're going to lose 33% of your clones, right? And that's a big hit to take when you're trying to find a needle in a haystack. So it's very important to consider how we are processing the cells and the plates in this workflow as well. Are we following a batch processing mode, or are there other options available? And there are, of course. There's serial processing of plates would essentially eliminate this issue. And what serial processing, what I'm referring to, is that instead of seeding all of your plates and then imaging all of your plates, what you would do in that workflow is seed a plate, image that plate, seed the second plate, image the second plate, so on and so forth. And now, instead of there being 4 hours between seeding and imaging, there'd be a maximum of 8 minutes that would pass between seeding or single cell isolation and image verification. And so you really wouldn't lose any clones. And that's what this graph here is trying to show you, is that a batch processing method is very sensitive to the time required to image plates, right? And the slower the imager, the worse batch processing performs. Whereas if you follow a serial processing workflow, you really won't lose any clones based on imaging speeds. And so it's not really a concern there. And now we have a product at Molecular Devices that we have developed in collaboration with a company called Cytena, called the Clone Select Single Cell Printer. And that allows for serial processing and overcomes many other limitations with the approaches to single cell sorting that have been described so far. Okay, so let me give a quick overview of the single cell printer. It uses, these products utilize microfluidic principles to create single cell containing droplets without the need for high pressures. So there's no complex oil interfaces or surface chemistries. It's really, very simple droplet creation mechanism. And that's going to increase cell viability and deposit efficiency while reducing the sample prep, contamination, and costs required. Okay, so how does this system work? On the left-hand side, you can see an image of the cartridge in which cells would be loaded. So pretty small there. The cells are then loaded onto the instrument. The cartridge is loaded onto the instrument. And in blue here, you can see depicted, this is where your cell suspension resides. And the system has a piston on it that is driven by a piezo, and that piezo-driven piston pushes against a membrane on the cartridge, which generates droplets. And those droplets that contain cells then are allowed to fall into the microplate. And what's important here is that cells never come in contact with the instrument, right? It's just the piston coming in contact with the cartridge membrane. And these are disposable, single use, right? And so the system is very little downtime, very simple to just swap out a new cartridge if you need to do a different cell line. You don't have all that decontamination that's necessary in a flow system. Okay, now a little bit more information about how single cells are deposited into the microplate will be provided on the next slide. But the way that it works is that there is a camera attached to the system, and that camera is capturing images of the cartridge region that would form a droplet. And if there's no if there's no cell detected within that region, then the droplet that's formed gets sucked off by a vacuum. Nothing gets deposited into the well. Now, if we move on to a scenario where a cell is detected in the nozzle region, that cell then would trigger the instrument to turn off the vacuum, which would allow the cell-containing droplet to fall into the microplate. In a scenario where more than one cell is found within this region, then the instrument would maintain the vacuum on, and that droplet would get sucked off as well, and so nothing would fall into the microplate. Okay, so only in this scenario does a droplet reach the microplate, and it should only contain a single cell. Okay, now what this allows for then is, going back to what we were talking about previously, is this allows for serial processing made possible by the fact that there's very little downtime, and also these additional factors which are mentioned here on the slide, that the single cell printer can be placed into a standard hood next to an imager, and you can quickly move plates back and forth between the instruments, whereas with a flow cytometry core or fax instrument, you're often going to a specialized lab, maybe maybe they're not even within your building, and so it's not possible for you to move between the fax sorter and the imager if you're using a core or something of that sort. So that's one of the reasons why serial processing is made possible by the single cell printer, but it's also provides immediate feedback to the user. So that way, if things start to go wrong, you can immediately correct it and continue processing things serially, right? And it has that very simple sample and instrument prep as well, which is very important. Okay, so the advantages, again, of serial processing is that it removes the burden of imaging speed on the workflow. However, imaging is still an important part of this workflow for other reasons, as we've talked about earlier, in terms of giving a higher probability of clonality. Okay. And what the single cell printers provides in this regard is an additional image of a single cell, which really gives an extremely high probability of clonality. Okay, and so how does that work? Well, if we look at, there's a publication again by Genentech here that looked at the probability of clonality coming off of the single cell printers, and in this scenario, they published a probability of about 0.98 off of the single cell printer. Now, if you combine that then with an imager, a downstream imager like the CloneSelect imager, now in this case, I'm assuming 0.9. In this particular publication, they used an imager, a different imager, and they had a probability of around 0.94 in that publication. But if I just assume a probability of 0.9, you can see easily that the probability is over 99% certain that it's a monoclonal cell line. Now, the advantages here are that you're going to get increased efficiencies with the single cell printer as well. And so only 18 plates are actually required with the single cell printer, and I'll come back to that in a second. So not only do you get higher probability of clonality, 2 independent measures of that as well, giving you very high assurance. You also do it with much less throughput needs. Whereas a manual method here, of course, you can get somewhat high probability of clonality, but it's 63 plates then to screen 1,000 clones, much more throughput needed. Okay. Here's a little bit further information about more details on that study that I just shown, and I'm going to go ahead and skip ahead just for the sake of time here to the following slide, because this is the more important one. And that is, in this study, what they basically did is they took green and red cells, mixed them together, and then sorted them. And the expectation is that if it's a monoclonal well, that'll either be green or it'll be red. But if a well is, got more than one cell deposited into it, then it would be a mixture of green and red. And so with all of the wells that showed outgrowth, 2,200 of them, a large majority were either green or red, right? So high probability of clonality already. Now, the mixed population is, of course, where the concerns would be. There was 9 wells. And what's important to note is that in one of the wells out of these 9, in one of them, the single cell printer showed a single cell. Okay, so that's a problem because we know that it's green and red. So the single cell printer did not accurately identify the single cell, but the Celigo instrument that they used did show more than one cell. So that accurately identified the well as not monoclonal. And so this well would have been rejected because the imager showed more than one cell. Okay, so the Celigo caught the problem, the single cell printer did not. Now these 2 wells, the single cell printer showed more than one cell. And so the single cell printer caught the problem, whereas the Celigo instrument missed the problem, right? But regardless, these wells would be rejected because there was more than one cell showing up in the single cell printer. Now you may wonder, how could that happen? The single cell printer, as I described earlier, said that only single cell containing droplets would be deposited into the microplate. Well, that can happen in a couple of scenarios. One, it's possible that the image software doesn't detect the second cell that's in the droplet. But that's the nice thing about the instrument what we highlighted earlier, that immediate feedback, is that you have the visual, right? So even though the software didn't detect it, you can visually QC and see, oh, there were 2 cells in that cartridge nozzle. So I can reject it for that reason. Okay. The other reason is that sometimes a cell might fall right outside of the region of interest that indicates the droplet formation. And in that case, then the software is only detecting what's in the inner circle here. In the outer circle, if there's a second cell there, it might accidentally get, or it might just, randomly get drawn into the droplet and fall into the plate, right? The nice thing about the system is that you also get an image after the cells have been ejected. And so you can see that at the point prior to ejection, you can see the 2 cells. And then if you look at the image after they've been ejected and see that there's no cells there, it went from 2 to 0, that tells you that 2 must have been ejected, and you can throw out that well as well, right? And so that's what happened in this scenario. And then this, as should be expected, is our biggest bucket of mixed populations. And these 6 wells showed 2 cells in both the single cell printer and in the Celigo imager, right? So both correctly identified that these were non-monoclonal, okay? And then the final important point here is that would be the most troublesome scenario. This would be a scenario in which both the single cell printer and Celigo both thought that the well was monoclonal, but it turns out it was not monoclonal. There were no wells like that. And so that gives you an idea here of, and this is out of, again, 2,200 wells. This gives you an idea of, how high the probability of clonality is with this combined workflow. Okay. And so now, kind of going back to another advantage of the single cell printer is going back to a previous point that we were making about screening cells for viability. So we had mentioned that if we could screen cells at 50% viability and not have to wait until they recovered to 100% viability, we could shorten the timeline by 13 days. But that a fax workflow really didn't allow this because fax is stressful to the cells and it kills them. But because the single cell printer is gentle, using microfluidic principles, it doesn't induce that sort of cell stress and cell death that fax sorters do. And so what we've seen in our hands is that cell viability can be improved with these systems. Okay, so on the left-hand side here, here's some data collected from a single cell printer. These were a cell population that was not very healthy. It was around 60% viability prior to sorting. And we did a limiting dilution sort. And we saw outgrowth of about 60%, which is to be expected then based on the viability. However, we also sorted these cells with our F-site single cell printer, which can do fluorescence sorting. And these cells were loaded with calcein-AM dye. And these are the same cells, I should have clarified that these are the same cells that were used in the limiting dilution study. But what we see here is that the viability was much, much higher with the F-site compared to limiting dilution, indicating that we were able to select out the viable cells from the non-viable cells using a sorter, right? Which is, again, the theoretical application when using FACS, but was not achieved because of the stress of FACS sorters. So this was not entirely surprising, right? Okay. What we also see with the single cell printers then, on this point of gentle sorting, is that this is really dependent upon the cell line that's chosen, right? And so we had two different cell lines that were used in-house, and I'll focus your attention over here on the right-hand side because this is the more interesting data. That we sorted single cells using traditional limiting dilution, a FACS sorter, and our F-site single cell printer. And for our freestyle CHO-S cells, what we see is that with limiting dilution and FACS, we get really poor outgrowth. But with the F-site, we get really, really high outgrowth compared to these two methods, right? Over 10 times improvement. But when we look to our recombinant CHO DG44 cell line, we see that the FACS sorter was actually quite comparable to the F-site, right? And so in this scenario, we got very good outgrowth of with both of these instruments compared to the limiting dilution method. And so really, it depends on the cell line whether or not FACS is going to have a major impact on viability or not. And so just to kind of summarize, the improvement in viability and throughput that you get with single cell printers compared to some of the traditional methods. What this diagram is showing here in blue is viable clones. So these are, your wells of interest are in blue. In green would be your wells that have more than one cell, so they're not interesting for that reason. In orange is your dead cells, and then your white circles here indicate empty wells. And so the higher density of blue wells is desirable, and that's what you get with the single cell printers compared to limiting dilution. This is at 0.3 cells per well, and this is with a FACS assuming about a 25% outgrowth, but 100% single cell efficiency, which is also not always necessarily possible. Okay, so because you get much higher outgrowth with the single cell printer, you're then able to get much higher throughputs and screen many more clones in a given 8-hour workday. So now this is combining both the sorting method and the imaging speeds to get a sense of what throughputs can be achieved. So using limiting dilution and FACS system that at 2 minutes per plate, if you have a really fast imager, you can get through quite a few clones with limiting dilution and FACS. But as soon as you start to get to these higher or slower speeds on the imager, then you start seeing a drop off in the number of clones that can be screened. Okay, and this, it's often the case that really you want to be screening more than 1,000 clones because a lot of the things you're looking for are quite rare. Okay. So if we convert this into the number of plates required for a given throughput, right? In most cases, limiting dilution and FACS are going to be unmanageable. You're not going to be able to screen that many clones, right? And that's what the red dotted line indicates here. But with the single cell printer at the 1,000 clone level, 2,500 clone level, you will be able to manage that sort of level of throughput. Right. But at some point, you're going to reach a throughput limitation if you need to screen, more than 5,000 clones, for example. So in this graph, you can see here that, it's going to take more than 50 plates to screen 5,000 clones, no matter what method you use. And so there's a fundamental limit there, and so that you can achieve in a manual workday. And so really, there's a couple of options in terms of increasing throughput beyond just the 8-hour workday. And that can really be achieved through custom automation, which is shown here in option 1, or through turnkey automation. And that's something that we provide with our ClonePix 2 system, and there's other products on the market as well. So let me just talk real briefly about how the ClonePix system works. ClonePix really is just an alternative version of limiting dilution in terms of the underlying methodology. What makes it truly unique is that instead of doing limiting dilution into a 96 well plate in liquid media, what you use here instead is a semi-solid media. So the semi-solid media sort of acts as the barrier, the wall of the microplate, and acts as the barrier separating single cells. And so then you can pack in these single cells at much higher densities than you can in a 96 well plate, where the max density is 96. And what you can then add into that semi-solid media is detection reagents, which can be used to bind to the antibodies being secreted from the clones. And you can then use those detection agents to find your brightest colonies, which would indicate your highest producers. And again, this allows for much higher throughputs because they can be packed in much more densely. And what the ClonePix does then is it adds in, plate handling and some robotic cell picking and gentle cell processing in order to pick these clones into 96 well plates. And so you can screen 10,000 clones in any given run on this system. Okay. And so if we take a look at whether customer turnkey automation is right for cell line development, if you look at option 1, where you're automating limiting dilution, which is the most common approach because it's difficult to automate other methods, really you're limited in this example, I'm going to maximize our efficiencies as much as possible. So I'm going to use 384 well plates now instead of 96 that I've been talking about in the past. And I'm going to use 0.2 cells per well because that's going to give us a high enough probability of clonality at the highest density possible. And so in order to, if we wanted to screen, let's say 3,000 clones using this approach with a custom automation solution, it's going to require over 8 hours of time and 64 plates, right? And it's also going to require all of these different instruments that need to be maintained and sustained. Now, there's a lot of advantages to this approach as well, which is that you've got a lot of flexibility built into the system, right? But as an alternative, and we support these sorts of systems through our custom engineering team who can do integrations of all of our products and can integrate third-party robotic handlers and third-party incubation. So we've built out many of these systems. But as an alternative, we offer the ClonePix system as well, which gives you the ability to screen 10,000 clones using only 10 plates, and that requires less than 6 hours to do so, right? So you can get through everything even in a typical workday. This system also screens for productivity, so it really combines essentially all of these solutions into one product, right? And gives you a pretty high probability of clonality, right? Not as high as this one, but it's decently high. But now we've customized the ClonePix to include single cell images, whereas previously it did not include single cell images, and that's probably a topic for another talk. But needless to say that the probability of clonality then would increase significantly if we added an imager into this workflow, as we've discussed previously. So then the final thing that I'll mention here is that the upstream sorting method is also going to have an impact on the liquid handling needs for downstream titer screening, which is sort of illustrated in the previous slide. So what do I mean by that? Well, with the single cell printer, if you have a very high efficiency of clonal outgrowth, then most wells in your microplate, you would want to screen for titer or whatever quality attribute you're looking for. And in that case, a simple, benchtop liquid handler, such as the one shown here, may be sufficient to transfer to an ELISA plate or maybe an octet system, right? And you'll only lose a few wells or a few pipette tips as part of this process. So you're not wasting a whole lot of reagents in this process. And I did some calculations here. 1 plate worth of reagents would be wasted for every 12 plates that were seeded, right? Or if you're using, for example, an octet system, one set of 96 biosensors could read, a maximum of 1,000 clones. Now, if I move over to a FACS workflow, most of the wells do not contain a clone of interest. And so simply replicating these plates onto an octet or an ELISA is going to cause a lot of wells that are not of interest. And so many reagents would be wasted here, right? So 1 plate of reagents would be wasted for every 4 plates that were seeded, right? And in this case, you would need many more biosensors, so 3 sets of biosensors in order to screen the same number of clones. So instead, you're probably going to require a much more advanced liquid handling system because your efficiencies are so low with this FACS sorter, right? And so again, going back to the theme here is that as you're scaling up these cell line development workflows and making trade-offs and decisions between what sort of sorter you're going to use, what sort of imager you're using, right? It has impacts both upstream and downstream. And there's costs and time associated with those trade-offs that you're making. Okay. Now you can combine these workflows in various ways to gain additional benefits, right? And so we've seen examples where customers have combined a ClonePix, which gives you high throughput, maybe a little bit less probability of clonality, with a single cell printer, which gives very high probability of clonality, but a little less throughput, to really give you the advantages of both systems, that you get the high throughput and the high probability of clonality, right? And that's just one example. There's many different ways in which these products can be combined into different workflows to gain additional benefits. And we'd be happy to talk to you, as a provider of many different types of instruments and solutions to help you optimize and understand what your workflow needs are. And so I'll just end with a summary of our cell line development workflow and the many and varied solutions that we provide for molecular devices across that workflow. And also just say that our portfolio extends beyond just cell line development. We really try to offer solutions that holistically match your sort of synthetic biology workflows, or however you want to call that, this design, build, test, learn sort of scheme, where we offer the electronic lab notebook through IDBS, which allows you to help design and learn from your experimental data, but also from the procedures that you're using. And you built out your antibody libraries through limiting dilution and imaging, or single cell sorting, or ClonePix, and then test the products using our plate readers and high content imaging systems. So we really provide a portfolio of solutions that can help you scale out and scale up your cell line development workflows.